Welcome to the Bioinnovation Progress Report. Our project brings together a large, multidisciplinary team of co-investigators spanning expertise in artificial intelligence, bioinformatics, medicine, and data science. Together, we are working to harness the power of foundation models to unlock new insights from biological data and drive real-world impact across research and clinical applications. This project set out to answer a fundamental question: how can foundation models transform the way we understand and analyze biological data? Our work spans the full pipeline, from surveying how foundation models are built and deployed for biology, to exploring agentic AI frameworks, conducting a dedicated review of RNA foundation models, and ultimately developing our own in-house RNA language model. Alongside this, we have built an AI-enabled research portal to make these tools accessible to biologists directly. Everything we do is oriented toward one goal: turning the vast complexity of biological data into meaningful scientific and clinical insight. To address the central question of how to build a foundation model for biology, we conducted a comprehensive survey of the existing literature. In the following slides, I will describe the progress we have achieved to date, and I will describe the next steps to be undertaken to finalize this project. We began this project by organizing the Foundation Models for Biology workshop, which inspired the direction of our research. Biology today is generating enormous volumes of data across multiple modalities, from genomics and proteomics to transcriptomics and single-cell sequencing data. This convergence of diverse biological data sets spanning genomic sequences, proteomic profiles, transcriptomic expression patterns, single-cell measurements, and spatial tissue maps represents a true era of biological big data. Our core premise is that foundation models, which have already transformed natural language processing and computer vision, can be equally transformative when applied to this biological data, enabling breakthroughs in drug discovery, protein engineering, and synthetic biology. In this survey, we approach the challenge of building a foundation model as a 5-stage pipeline. We start with identifying and collecting big data in biology, then move to data preprocessing to make the data model ready. Next comes choosing the right architecture, followed by the pre-training phase, where the model learns general biological representations. The final stage is adaptation, where the pre-trained model is fine-tuned for specific biological tasks. This structured pipeline gives us a clear lens through which to evaluate and compare existing models in the field. Let's look at the first stage, big data. We systematically categorize biological big data into 5 major categories. For each category, we identified and mapped the number of distinct data sets, what we call data islands. This mapping is important because it reveals where data is abundant and where gaps still exist, which in turn guides decisions about which biological tasks are ready for foundation model approaches. Once data has been identified, the next step is pre-processing. Raw biological data is rarely suitable for direct model training. It often contains noise, missing values, and inconsistencies. In our survey, we identified 5 key pre-processing techniques applied in the literature to transform raw biological data into model-ready formats. These techniques are critical for ensuring that models learn meaningful biological representations rather than artifacts of the data collection process. Now we turn to model architecture, the structural design choices that determine how a foundation model processes biological information. We categorize architectures along 2 dimensions. The first is the type of computational module: convolution for extracting local features from nearby sequence positions, attention for capturing long-range global dependencies, hyena for handling very long sequences efficiently, and state space models for sequential processing of distant sequence units. The second dimension is the overall structural pattern: encoder-only models for understanding the sequence, decoder-only models for sequence generation, and encoder-decoder models for sequence-to-sequence tasks such as translation. After choosing an architecture, the model must be pre-trained. Pre-training is what gives foundation models their power. It allows them to learn general-purpose biological representations from large amounts of unlabeled data. In our survey, we identified 3 main pre-training objectives: mass language modeling, which trains the model to predict hidden tokens and is excellent for building deep sequence understanding; next token prediction, which is used in generative models that produce biological sequences, and denoising objectives, which train the model to reconstruct corrupted inputs and tend to be more robust in the presence of noisy biological data. The final stage of the pipeline is adaptation, taking a pre-trained foundation model and specializing it for a specific biological task. In our survey, we organize adaptation strategies based on two factors: the type of input data the adapted model receives and the downstream task it is being applied to. This categorization helps researchers identify which pre-trained model and adaptation strategy is best suited for their specific application, whether that is gene function prediction, protein structure analysis, or drug response prediction. Beyond building and adapting foundation models, our survey also explores a more advanced paradigm, agentic AI. You may recall our dedicated agentic AI workshop, where we introduced what agentic systems are, when they are appropriate, and how they differ from conventional model training. In this section of our survey, we investigate how agentic AI is being applied to biological big data processing. We have categorized existing agentic biological systems into 4 groups, each representing a different level of novelty. The slide shows a selection of notable systems, including Virtual Lab, BioResearcher, Stellar, Gene Agent, CRISPR-GPT, and many others. Complementing our survey work, we have also built a practical demonstration of agentic AI in action through our AI-enabled research portal. You might remember the workshop we organized earlier this year to introduce this portal. During this workshop, we showcased a system that integrates multiple well-known biological AI agents into a single unified interface. This portal is designed to make cutting-edge biological AI tools accessible to researchers without requiring deep technical expertise. If you are interested in accessing the portal or contributing additional tools, you can do so by scanning the QR code shown on this slide. To give you a sense of the depth of our survey, this slide presents some sample tables from the paper. These tables summarize and compare the foundation models we reviewed across key dimensions such as big data explanation model architecture, pre-training data, and agentic AI. The structured tabular format makes it straightforward for researchers to identify the most appropriate model for their specific biological use case. While our broad survey covers the full landscape of biological big data, we recognize the importance of going deeper in one specific modality. Within the vast space of biological data, we chose to focus our original research on transcriptomic data, more specifically RNA sequences. RNA sits at the heart of gene expression and regulation, yet it has received comparatively less attention in the foundation model space than DNA or proteins. This became our primary research target going forward. Now I discuss our second major contribution, a dedicated survey on RNA foundation models. While general biological foundation model surveys exist, we identified a clear gap in the literature for a focused, systematic review of models specifically designed for RNA. Our RNA Foundation Models Survey makes 2 key contributions. First, it is the most comprehensive review to date, covering 46 RNA Foundation model papers, significantly more than any previous survey in this space. Second, we go beyond RNA native models to investigate how foundation models trained on other biological modalities, such as DNA or proteins, can be adapted for RNA processing through cross-modality transfer. Turning to the data used to train RNA foundation models, we found that both coding RNA, which encodes proteins, and non-coding RNA, which plays critical regulatory and structural roles, have been used as training inputs. The figure on the left shows how many of the 46 models we reviewed rely on each specific RNA dataset. This visualization reveals which datasets have become de facto standards in the field and which RNA subtypes remain underrepresented in current foundation model training. If you remember, last year we organized a crash course on AI for biologists, during which we presented some examples of foundation models for RNA, including Rinalmo. In our second survey, we compare all 46 RNA foundation models based on their architectural choices, overall model size, and the computational resources each uses. A key observation is that larger models generally require substantially more computational resources to train and run. As a concrete example, Rinalmo, which serves as a basis for our own in-house model, is among the largest models in this survey and accordingly demands significant hardware infrastructure to work with. In the pre-training analysis of our RNA survey, we categorize models along two important dimensions. The first is the tokenization technique, that is, how the RNA sequence is broken down into input units that the model can process. The second is the pre-training objective, the specific learning task the model is trained to perform on unlabeled data. Understanding both dimensions is critical as they fundamentally shape what biological knowledge the model internalizes and how well those representations transfer to downstream RNA biology tasks. For downstream adaptation, we organize the tasks that RNA foundation models have been applied to into three broad groups. This categorization provides researchers with a clear map of the current application landscape for RNA AI, spanning sequence analysis, structure prediction, functional annotation, and beyond. Researchers can use this framework to identify the most appropriate pre-trained model and fine-tuning strategy for their specific RNA biology research question. We now move to the third major component of our project, the development of our own in-house RNA language model. Building a large-scale foundation model from scratch involves substantial engineering challenges, and this slide presents an honest account of the obstacles we have encountered and the solutions we have implemented. The most significant challenge has been computational resources. On the data management front, we implemented clustering-based pre-processing, following the original Rinalmo paper's recommendations to increase training data diversity. An important distinction we have carefully studied is the difference between inference and training workloads for large RNA language models. Training requires sustained access to large GPU memory and compute over extended periods, whereas inference, running the trained model to make predictions, can be executed far more efficiently using optimized deployment strategies. Understanding this trade-off is essential for planning our resource usage throughout the project and for eventually making the model accessible to the broader research community. Finally, let us look at where we stand today with our in-house RNA language model. We have commenced pre-training, and this slide shows three key training metrics measured at approximately 30,000 iterations. The loss curve on the left shows the model's prediction error decreasing steadily over time, exactly the behavior we expect from a well-converging model. The perplexity plot in the middle confirms that the model is progressively improving its understanding of RNA sequence patterns. The rightmost plot shows our data throughput, currently processing around 36 samples per second. We are making solid progress and remain on track with the project timeline running through December 2026. Before we close, I want to briefly reflect on our journey. On the positive side, the rapid rise of agentic AI expanded our survey in ways we did not anticipate. We also discovered that models trained on DNA and protein data transfer surprisingly well to RNA tasks, a finding that became one of our key contributions. In terms of challenges, computational resources remain our biggest constraint, and managing biological data at foundation model scale required us to adopt more sophisticated approaches than originally planned. Finally, bridging the gap between AI researchers and biologists required ongoing communication and encouragement. Looking ahead, if further funding became available, our immediate priorities would be scaling our RNA language model with greater compute and larger datasets, and fine-tuning it for a specific disease context, such as motor neuron disease, in collaboration with our biologists at Macquarie. This project has also opened some exciting doors. We have 2 survey papers in development, strong potential for ARC discovery and NHMRC funding applications, and growing collaborations with the biology community. We have also developed a talented cohort of students with rare combined expertise in AI and bioinformatics, a real asset for where this research is heading. Before concluding, I would like to take a moment to express my sincere gratitude to my talented students who have contributed to this project as research assistants. Your dedication, enthusiasm, and hard work have been invaluable to our progress. Thank you. To conclude, we are actively collaborating with biologists at Macquarie University to identify a focused application domain, such as motor neuron disease, with the goal of developing a dedicated RNA language model tailored to that disease context. Thank you for your attention, and I welcome any questions you may have.